Welcome at the BIND community webinar for the patient community. My name is Susie Ann Baker. I'm from the World Duchenne Organization, and I will be your host for today. So this is just a brief overview of the agenda today. We will start with an introduction of the BIND project by Professor Francesco Muntoni, and then we will move on to what we have learned throughout the project. So what we've learned from mouse models, what we've learned from MRI scans we've made in the brain with Duchenne and Becker. We're also talking about how to assess cognitive and behavioral challenges in Duchenne and Becker. Before moving on to the outcomes and next steps and the moderated questions and answers. So with nothing further ado, I'd like to hand over the talking stick to Professor Francesco Muntoni, who's a pediatric neurologist and the BIND project coordinator. Thank you very much.
Thank you for arranging this. I'm just, yes, should be able to share the screen. Well, firstly, thank to you, Susan, and to the World Duchenne Organization and the DDF, because the support you have given to the project is unprecedented. And the work that you continue to do with us for taking forward the work of the consortium is absolutely essential. So I am a pediatric neurologist. I've been lucky to be the coordinator of the BIND project that started in 2000, finished in end of June 2024. So in actual fact, we are still digesting much of the information that is coming from especially the clinical aspect of the project. The point of this slide is that only some individuals with Duchenne, but also Becker, have cognitive or behavioral issues. The question that the BIND consortium had set up to address is, do we understand why some people and not others have these comorbidities, and what can be done about that? And I think, as you can see below, dystrophin has, of course, a very good role and very well understood role in muscle and heart. That is what we are all working towards for novel therapies. However, there are dystrophin isoforms also produced in the brain. And while all people with Duchenne lack muscle dystrophin, that isoform is also present at low levels in brain. And depending on where the mutation is in the dystrophin gene, also other isoforms can be affected. Next slide, please. So in order to address this and other questions, we obtained funding for the European Consortium to address a number of issues that go from basic science that we're not going to discuss today to more clinically applied science in terms of the various aspects of people with Duchenne muscular dystrophy, also animal models of Duchenne muscular dystrophy. Part of the BIND consortium have been involved in trying to identify genetic therapies so that dystrophin restoration could occur not only in muscle, but also the brain. But this meeting will be mostly focused on some of the clinical achievement of our consortium. Next slide, please. So this gives you explains that the top where there is the red arrow, the large red arrow, that is the full-length dystrophin isoform that, as you can see at the right-hand side, is present in muscle. This is the very well-known normal expression of dystrophin that will be absent in people with Duchenne. But then you can see from the right-hand side of the same larger isoform, there is a red arrow indicating that that isoform is also expressed in brain. Thank you very much. And then there are two more red isoforms indicated as DP140 and DP71. And these are also expressed in brain, and it is the combined deficiency of one or two or three of these isoforms that is important for the difference in the prevalence and to some extent severity that is encountered in people with Duchenne. Next slide, please. So what progress has been done so far. I think that we will not discuss about this, but we have, for the first time, look very precisely which isoform is present where, in which cells, and also aspects that are, for the families that are much more aware of the role of dystrophin in muscle, that that protein is associated with a number of protein called dystrophin-associated glycoprotein complex. And we have now evidence of some of these protein complexes in the brain that are different from those in muscle. Next slide, please. I think we have, and colleagues mostly in France have identified a clear behavioral profile in mice deficient in the different isoforms. So we can differentiate mice that are different, affected, that have involvement of multiple or only one of these isoforms. As I indicated before, we have used genetic therapies, mostly antisense oligonucleotides, similar to the techniques used for muscle, but also some gene therapy to improve outcome in mice. And I think that our results do suggest that, at least in mouse models, improvement is possible. And of course, that means that in the future, this could be pursued. Next slide, please. I think you will hear mostly from Jos now, so I will keep this very brief. How in the human, what happens in people with Duchenne and Becker's, and once you have assessed this also, can some of this information in the future be used if we wanted in a few years' time to run clinical trials focus on dystrophin restoration also in the brain? We need to have clear outcome measures, very similar to what we use for muscle, the North Star, the 6-minute walk test. But if we wanted to do something for brain, we do need to know what we may be measuring. This may be my last slide. Next. And I think, yes, I think this is, I will discuss this afterwards. So I try to keep the focus of this few slides on the clinical development, only a very high level of the preclinical development, because the focus of this webinar today is really mostly where we are in terms of our clinical development. So I will stop here so that the next speaker has his or her chance.
Thank you very much for the introduction to this project, Professor Montoni. Now we move on after the introduction of the BIND project to the what we've learned from the different work packages that is chaired by Elizabeth. So Elizabeth, the floor is yours to introduce the speakers.
Thank you so much. And I think this is also a right moment to thank Professor Montoni for taking the initiative of this project. And I know this has been a burden for many years, a burden in terms of it's a lot of work, but this is really something the Duchenne community was waiting for and are very grateful that you took the initiative. So after I said that, I will introduce the next speaker, which is Dr. Amel Saoudi. She works together with Dr. Cyril Vaillant in the French National Science Institute, which is called the CNRS, as you can see, the acronym is behind her name. And they've been looking into mouse models. And the floor is yours, Amel.
Thank you very much. I'm just going to share my screen. So yes, I'm Amel Saoudi, and I've been working with Dr. Cyril Vaillant in France. So I was involved in the BIND project for a couple of years during my PhD, and we've been working on various mouse models that were missing specific brain dystrophins in order to better understand how these proteins affect the behavior, the memory, and the movements in these mouse models. So we had the main questions. So as an introduction, we had 3 main questions. The first was to identify and understand the cognitive, behavioral, and neuropsychiatric aspects in these DMD mouse models. And then we wonder whether the mutation position, so the mutation position, and thus the number of dystrophin impacted, the dystrophin loss, impact the deficits profiles. And all this work is in order to specifically provide a set of reliable behavioral tests to evaluate further therapeutic studies. So as I was saying, we were using different mouse models for Duchenne muscular dystrophy. And these models has different mutations in a different position on the gene and lost different brain dystrophins. So the first mouse model is named the MDX5CV mouse that has a mutation at the very beginning of the gene and loses one dystrophin, the DP427, in the brain. The second mouse model is the MDX52 mouse that has a mutation as in the middle of the gene and loses two different dystrophins in the brain. And this is the most frequent profile that is found in 60% of patients. And finally, the last model is the DMD null mice that carries a mutation that mimics the loss of three different brain dystrophins, the DP427, DP140, and DP71, and this represents less than 10% of the patient, but shows the most severe aspect for the central nervous system. So we had a large strategy to compare these three mouse models that has three distinct mutations, and there were more than 28 different behavioral tests that were replicated and completed in three different laboratories within the BIND project, in which we included different functional studies that assess the motor aspect, the social interaction, emotional reactivity, and so on. And the aim was to determine specifically a behavioral toolbox to assess treatment efficacy in further studies. So I'm showing our main results in these behavioral studies, starting with the stress reactivity and the anxiety. So to assess the stress reactivity in mice, we hold them in our hands and then put them back in their cage, and we measure the immobility of the animals following this manipulation. And the immobility shows the amount of stress reactivity of the mice. And here we can see in different colors, blue, red, and green, the different DMD mouse models. And we see that they have a very high immobility, which indicate that they do have an increased stress reactivity, and that this phenotype is comparable across the three different mouse models and is robust and replicable, which could be a solid outcome for further studies. Also, it also has been shown that this phenotype has an age-dependent progression and develops mainly during adolescence. Regarding the anxiety, we had three different tests to assess it based on anxiogenic conditions for the mice, which could be very brightly lit compartments or also the fear of the void. And in this part of the study, we showed that the different mouse models, the three different mouse models, has a anxiogenic phenotype, and this phenotype was more severe in the mouse models that has multiple and different dystrophins missing in the brain, which shows that the severity is dependent on the mutation. We also could observe that this phenotype is age sensitive, as it is more severe in adolescents, and it may vary depending on the genetic or the familial background. Going further in the emotions, we looked at the emotional learning of the mice, and we observed that the emotional learning was delayed in the three different DMD models, and also the memory was impacted. And this phenotype was very robust and replicable, but also it was comparable between all the models. These kind of studies also let us know which brain region is affected and involved. And here is the amygdala, which is a brain region known to be the center of the emotional memory. Further in the learning, we looked at the spatial orientation, so the ability to spatially orientate, and we could observe that in the different DMD models, we had an impairment in the spatial orientation, and this alteration was more marked in the animals that has a mutation in the middle or the end of the gene, which could indicate that the protein involved in this behavior is the DP140. And also we could say that the brain region that is impacted is the hippocampus, which is a structure in the brain that is the center of spatial orientation. We went further in the learning and looked at the flexible behavior, which is the capacity to adapt to a variation during a learning task. So the animals are submitted to a learning task, and then we change the rules and see how they adapt and relearn the task. And we observed that this adaptability of the animals was impacted in the mouse models with the mutation at the middle and the end of the gene, which is once again dependent on the DP140. And this phenotype was strong in adults, so it was reproducible. And the structure involved, so the brain structure involved, is the prefrontal cortex that is impacted, as it is known to be a center for the flexible behaviors. And finally, we also looked at the social behaviors. And for the social behaviors, the different studies have been conducted, and we now know that the DP427 slightly affect the loss of DP427, slightly affect the social behavior, and the loss of the two different dystrophins in the brain, or three, could also slightly affect, but in a different way than the loss of one brain dystrophin. And current ongoing study in the lab is also working on the influence of the emotional disturbances on the social behaviors. So overall, we answered the questions asked at the beginning of this talk, which is like, so far we know that we have a characterization of the cognitive, behavioral, and neuropsychiatric aspects in these DMD mouse models. And also we showed the impact of the mutation position, but also the number of lost dystrophin in these phenotypes. And overall, we know that the DP427, the loss of DP427 enhances the anxiety, the stress reactivity, and impairs the emotional learning. The loss of DP140 worsens the anxiety, impairs the spatial orientation, and alters the social behavior. And finally, the loss of DP140 and DP71 reduces adaptability and impacts the interest on social novelty. And all these studies and all these different tests allowed us to specifically identify a behavioral toolbox of behaviors that were solid and replicable to further assess the treatment's efficacy in further studies. Thank you.
Thank you, Dr. Amal Saoudi. Thank you so much for this presentation. And to the audience, there is a Q&A. You see it. Oh, there's a Q&A. So you can ask questions in the Q&A, which can be answered later. So don't hesitate to put your questions there. So thank you so much. We continue with the next speaker, who's Dr. Hermine Kan from Leiden University Medical Center. She will show the results of the MRI studies in the patients, and the floor is yours.
So thank you for the opportunity to talk here today. I'll also start my timer so I don't take too long. So I will share some of the results, but mainly actually most of the plans that we did using MRI scans of the brain in Duchenne, in patients with Duchenne and Becker muscular dystrophy. So I wanted to share with you what actually MRI is. So MRI is my favorite imaging modality. I spent most of my research career using this modality, but what is it actually? And why is it called magnetic resonance imaging? Well, an MRI scanner is actually a very strong, like a tunnel with a very big magnetic field that you see here. And to give you some numbers, the scanner operates at 3 Tesla, and 3 Tesla is actually the same as 52,000 times the Earth magnetic field. So it's a pretty strong scanner, pretty strong magnet. So now I press the image and now it's going wrong. Yes. So what else do we need? We also need an antenna to receive the signal. So those of you who have been in a scanner before, you will lie in a contraption like this if we want to scan your brain, or there's a contraption like over your chest if we want to scan the heart. And this is because the signals that are emitted from the body are actually really tiny, and we want to be able to receive those. And this is the actually antenna that we use to receive that signal. And we only receive signals that are in resonance. So that's why it's called magnetic resonance imaging. So the other thing that we need is headphones, because the magnet makes a lot of noise. To be able to obtain the signals and to protect our ears against that noise, we can wear headphones, or usually when we scan children, but also adults, we also offer them that they can listen to music or they can watch a video. So what can we do with MRI? Well, we can look inside the brain, and that's what I think is so cool. We can actually have a look what is going on here. So what do we see here? We see the skull on the outside. We see the subcutaneous fat under the skin. We see a nose here. We see eyes. So this is a transfer section through the brain. So basically we sliced it like this. We can see the ears here on the side, and here we can see the brain. And the brain is what we've been studying. And in the brain, there's gray matter. So that's the grayish stuff here. And there's white matter. And the gray matter is basically all the cells that we use to think and to do all the other things in the brain. And the white matter is basically the highways of the brain. That's how the signals get get moved from one part of the brain to the other. And another cool thing about MRI is that you can actually do different types of scans. So if we put somebody in the scanner and they lie still for long enough, we can actually do different types of measurements. And I'll tell you a little bit about the things that we did in BIND and what we can learn from them. So first thing that we did is to perform what we call structural MRI. And with structural MRI, we look at the structure of the brain. So we look at gray matter and white matter. I told you a little bit about that before, right? The gray matter are the cells and the white matter are the tracts, are the highways. And we can also look at the volume of the brain. And we do this in so-called T1 weighted images. And this is a T1 weighted image of a control. And this is a T1 weighted image of a boy with Duchenne muscular dystrophy. And at first glance, there's not a really big difference between the two. But when we really went into the details in earlier studies, and we did that, and also other people did that, and what we found is that the gray matter, so the number of cells basically is reduced in patients with Duchenne muscular dystrophy. This reduction was stronger. Colleagues from Leuven showed that when patients take daily deflazacort compared to 10 days on, 10 days off prednisolone. But these type of scans have never been done in Becker muscular dystrophy, so there we don't know what's going to happen. So another scan we can do is DTI, and we summarize it basically with structural connectivity. That's how we call it. And this is basically the highways of the brain. So if you imagine different cities that are connected in a country, there are highways between those cities, and those highways basically enable cars to drive from one city to another. But that's basically also what we measure with DTI. So this is zoomed in. So this is a nerve cell, a neuron, which has long dendrites. So these are the highways basically. And they're like little cells surrounded. And what we do with DTI is we actually measure the movement of the water molecules. And what you can see that if a water molecule is here in free space, it can move a lot, right? So that means we get a certain amount of signal. But if a water molecule is within a cell body or is even within one of these long dendrites, it will move differently. And we basically use this property to make diffusion tensor imaging, so DTI, to highlight where these highways are, because the water moves differently in the highway compared to within the city, and that's why we can make a certain image of it. So what we've done in the past, we looked at this in patients with Duchenne muscular dystrophy, and we saw that it was altered. And again, in BMD, we don't know what's going to happen. So this is a type of image like that. So this is a raw image, and then in an analyzed image like this, you get the different colors, which basically means the main direction that the water is moving. So blue is for instance, from left to right, and red is from left to right, and this could be green from back to front, for instance. So another thing we could measure is the perfusion. So perfusion basically means how much blood is flowing through the brain, right? Our brain is really well perfused. We have a lot of blood vessels going on, and we can use a different type of scans to basically measure how much blood is going there. And if you remember a little bit from the previous images, we can see that there were a lot of highways, so a lot of white matter, white tissue of the brain, while the cells are more here to the outside. And you can also see that there's more blood going to the outside and going to the nerve cells, basically, rather than to the highways. Again, we've already looked at this in smaller studies in Duchenne, where we've shown that in Duchenne, in patients with Duchenne muscular dystrophy, it's reduced, while in Becker muscular dystrophy patients, we don't know. So the last type of scans we looked at is called functional MRI. And with functional MRI, we actually look at brain activity. So I told you that there's a lot of blood going through the brain, flowing through the brain. But what we do with functional MRI is we actually look where the blood is flowing. And we ask somebody, for instance, to look at a checkerboard, a flashing checkerboard, or we ask somebody to perform a task. And then we look at different regions of the brain where that is becoming active at that moment. Because if the brain is becoming active, there's a lot of blood going there, and we can actually see in the brain on these type of scans that you see here with the red dots, with the red areas, that there's a lot of blood going to that area of the brain. Again, previous results in Duchenne show that this was altered, and we don't know what's going on in Becker. So what did we do now? So we did in BIND, we did perform both human studies and preclinical studies, so the mouse studies that were just explained, and in both of the species, so-called, so both in human and in mouse, we looked at structure. So we looked at the amount of cells, basically at volume. We looked at structural connectivity. So we looked at the highways. We looked at perfusion, so how much blood is going through the brain. And we looked at functional MRI. So another novelty in BIND is actually that we did this project with a lot of different sites. So we scanned patients in Copenhagen, in Rome, in Leiden, in London, and in Newcastle. And even though MRI is my favorite modality, not every MRI scanner will give you the same results. So what we actually had to check first before we could start analyzing the data is to see if we let the same people travel to different sites. So we asked healthy volunteers to travel between these different sites and do the same type of scan. And do we actually get the same results? So the nice thing is that we do. So we asked 6 people here. So these are the travel heads here at the bottom, 6 people to travel to all the different sites. We measured their total brain volume. And even though they, of course, differ between the volunteers, so Travelhead 3 has the lowest total brain volume and Travelhead 1 has the largest, you can see that these dots are actually really close together. So if you measure the same volunteer at a different type of scanner, you will get a very comparable result. So that's really good news if you want to start analyzing the patient data. So a little bit about inclusion rates. So we scanned Duchenne patients, Duchenne controls, we scanned patients with Becker muscular dystrophy, and controls for the Becker muscular dystrophy patients. So these are all age matched, and we scanned travel heads. So you can see that the inclusion rates are quite high, right? So these are all the patients that we scanned in total. So we're really happy with that. I guess that's the good news, because the bad news is that we only finished scanning these patients in April last year. So I don't have a lot of results to show you, but we have the data in, so that's the best news, I guess. So what is now new in BIND compared to what I showed you earlier? Well, I mentioned that there were earlier results, but the link between mouse data and human data has not been made. So that's something that we will do. In Becker, there's not a lot of MRI data at all out there, so that's also what we will do. We will look both in Becker and in Duchenne at advanced structural connectivity. So we can do even more sophisticated analyses than we did in the past. The same holds true for fMRI. And finally, I think one of the most important things is what we will be able to do is to do the link between MRI and the neuropsychology and behavioral tests that Jos Hendriksen will share later. So what are our next steps? Well, the mouse data is basically, we've analyzed everything and those are ready for publication. The human data have been checked, the quality is good, and we can really now start analyzing those. And in the next 2 months, we will perform the MRI analysis, we will add the data to the DDF platform, we will link the data with neuropsychology and behavior and publish and disseminate the results. And the good news is that we've obtained funding from different parent projects to actually do this work. So in the next year, there will be more news coming from us. Thank you.
Thank you so much, Dr. Kang. And I already see questions coming in, but we will do this later. And then we will go on to the next speaker, also from the Netherlands, Dr. Jos Hendriksen from Kempenhagen.
Thank you. I share my screen. Hope it works. Well, thank you for the opportunity to give a short presentation on the assessment of cognition and mental health in Duchenne and Becker. I prepared this presentation together with Ruben Miranda from Madrid University. And we did this work on behalf of a working group of psychologists and neurologists from the 7 participating centers in the BIND study. You have all the names here, and we present these data on behalf of this whole working group. I will present in the next few minutes 4 topics to guide you through what we know a little bit of and what is the work in progress. I will first tell a little bit about the theoretical framework. Then I will tell a little bit how we assessed cognition and mental health in patients with Duchenne and Becker. I show 2 results of all the results that will be coming. And I will finalize with a short presentation of a screener, which we also developed as part of this BIND project. The first topic is about the theoretical framework. When you read the literature, you can say that there are around 10 areas of brain-related comorbidities which have been described in patients with Duchenne and Becker, for which they are at a higher risk. And we call it the Big 10. And if you look at that model, it could be like this. We have 4 domains, as you can see. We have the domain of cognitive functions, which is the A domain. The second domain, the B domain, is about the learning issues, the reading and the arithmetics. There is a third domain covering 2 areas, anxiety and depression, which gain a lot of interest in the literature. And the 4th domain is the D domain, which is about neuropsychiatric problems that have been described in literature. And when you look at this picture, this is a simplified picture, because when you look at clinical practice, we know that there is diversity of problems. And we know, and our data, the BIND data, also show that there is a complex overlap. And it is difficult to have a clear, uniform diagnosis. And this needs a lot of work, because this is what parents worry about. Is it autism or not? But probably we have to say that there is a combination of problems. So this neurodiversity will hopefully be clarified a little bit with the data that will be presented and will be available after the BIND study. That brings me to the second topic, because that has to do with assessment of mental health and cognition. What we did as part of the BIND study is that we looked at psychological test usage in all the participating EU multi-centers of the BIND study. And what you can see in the middle column here for the age from 6 to 18 years, when we asked the 7 centers what kind of instruments they use, there is an amazing different of instruments being used. For intelligence and cognition, there were 38 instruments, and for behavior and psychiatric problems, there were 26 instruments, different instruments used by different centers. So that makes assessment in a harmonized way a very difficult issue, and this has to be tackled. So that is why we tried in the BIND consortium and in the work package to get a core set of variables. And you see here what we, how we try to harmonize the data which we have collected in the BIND and which are going to be analyzed, because the final data have been collected a few months ago. And the data are about behavior, and they are about cognition and academics. And these are areas of the Big 10, which I talked previously. The behavior has been assessed by a questionnaire and by an online interview, and we call it part 1. So it was parents who answer questions and have an online interview, and this gives information on the behavioral issues. And the cognition and the academics were also tested in an in-person psychological assessment of patients with Becker and with Duchenne in all 7 centers. And these data we had to harmonize. And this is one of the publications which is currently ready and has been submitted to neuromuscular disorders. It's a workshop report about 4 expert workshops, which we had in Newcastle, Leiden, and in Rome and in Paris, to harmonize the clinical tools for assessment of brain-related comorbidities. So there is work to be done, but we have had some work, and we had a lot of work about harmonizing all the clinical tools. This brings me to the third thing, which I want to talk a little bit, to give you a sneak preview of some preliminary results. But I want to ask you not to copy these data because they are still subject to correction and they are still have to be published and they are still work in progress. But these will highlight what we have on the data. Well, to finalize, there is some work in progress. We can say that we have the biggest sample of behavior and cognitive data together in one DMD-MB sample collected in an harmonized way. So we have Duchenne data of 277 patients, 36 young Becker patients, and 51 adult patients. And with these data, we can develop a Duchenne or Becker specific neurocognitive test kits. And of course, what Hermine Kan said, it is important to link mice data and MRI data to the human data and to find out how we can learn from each other. Thank you for your attention. This is my presentation.
So thank you so much. And before going to the next speaker, which is Professor Muntoni again with the next steps and outcomes, is if the previous speakers can have a look at the Q&A and see if there are some questions they already can answer in writing, because we only have less than 10 minutes to ask them in person. Maybe there's some they prefer to answer in person, but on the other hand, just in writing, if that is feasible, would be really fantastic. Thank you so much. And now the floor is for Professor Muntoni again.
Right, thank you very much. I think that the I cannot speak and type. I saw some question that I could address, and I'll do my best to do that. So the next slide, please. And thank you, Susie, for helping. So what have we done so far? I think we have already, if colleagues and families are interested, there are already 19 papers that have been, that you can find on the BIND website together with lay summaries. We are working at least another 20. Probably this is the estimate will be higher. And I think for some of you, and both Hermine and Jos, who I thank again for the work, have mentioned that some of the work is not completed. The reason for this is that there has been COVID, you maybe remember, and the analysis, if you like, everything has been shifted towards the very end of the project. So I think that is one of the reasons why we haven't completed the analysis of the clinical data. I think the other important thing for you to know is that the data of the BIND have been deposited with the Duchenne Data Foundation, and this will be available also outside investigators, outside BIND for future, in Ashgabat forever. Next slide, please. Other things we're doing currently is we have realized, and I think you have seen from Jos, the level of pharmacotherapy is not great in many countries, including the one where I work. So we want to improve this aspect, both in terms of the assessment and also agreement on the pharmacotherapy to be used. Next slide, please. We have already started to work with pharmaceutical companies regarding what did we find. Can we partner with them regarding some of the clinical data, for example, in terms of who has the knowledge to power and to perform clinical studies and what to expect in people with Duchenne. When you target in the brain, we will be able to help them in this area. Next slide. And at the same time, it is important. Different therapeutic strategies may have different profile potential adverse event, different mode of administration. And while we are not ready to propose that we have tomorrow a therapy, I think it is useful to start to discuss in focus group with the families. This is, Elizabeth has already facilitated one of these meetings, and it was very, very interesting, what is called the patient preference studies. And this will take a little while for us to have full assessment of this. The last slide is, you already heard that Leiden has obtained some funding to continue to analyze the data from obtained during the BIND. The last scan was in April this year, not last year. So that is why there has not been time to analyze it. So Leiden will now have time to do that. I'm also grateful for the World Duchenne organization, because we have a 2-year part-time coordinator to really pursue, together with all the BIND partners and the EDF, the possibility of a legacy structure. And of course, each of us is involved in ongoing grant application. I just defended a grant application of myself this morning on exactly area related to the BIND consortium. This is my last slide. And so I pass on the microphone to Elizabeth.
Thank you so much. Thank you so much. And then I think we will together go and have a look at the question and answers. Okay, then with this, I would like to thank all the speakers. It was really amazing. And also want to thank you for all you do and have been doing in the last 5 years, let's say, right? In spite of COVID, all the difficulties, you have done a lot of work. And so thank you so much for tonight. We will, with this, go to the next speaker, which is, again, I think, Dr. Muntoni, right?
I was just going to say, well, my task is very easy, because I think you thank us, Elizabeth. I need to thank you. I think that certainly the focus that the families and the fact that you and the advocacy group have been able to fertilize the work that we are doing, and hopefully, as we know, in actual fact, we think the BIND we haven't sorted everything. We have clearer various aspects that now we will address. We have certainly achieved much, but I think that we now need to focus on some of the unmet needs. With these clearer in our mind, and I think in partnership with all the investigators in BIND, new investigator, and the advocacy group, I think we can certainly put BIND and the brain in Duchenne and Becker under the spotlight. Thank you very much to everybody.
Okay, thank you so much also now to the participants, because without participants, the webinar has not a lot of views. So thank you so much. I wish you all a good evening, and hope to see you in the next webinar.